Okay, so first of all, I would like you to pass this page. Just write your names, and pass it along. So, yeah, you can write it in Hebrew also. It doesn't really matter. What? Yeah, but I understand also Hebrew, so yeah. Yeah, but you can also write in Spanish, which is very okay. So, on. Can you close the? It's funny how you're Saturday. Everybody is on this side. You're like, yeah. Interesting. Okay. So, okay. So, last time, actually, when I went over it this time, I realized we didn't do so much last time. But so, what we did do is we started talking about molecular motors, and I showed you basically we went into two examples of molecular motors. One of them was the action of the muscles, where we saw the myosin and the dynein and the way that they function. Specifically, we divided it into steps, and like I was asked here before, a lot of time our division into steps is a very arbitrary thing because everything happens in a continuous manner. So you need to imagine it as a process that is happening continuously and in many sites of the cells. But basically what's important here to understand is that the function of the molecular motors is pretty simple. What they actually do is that they convert chemical energy in the form of ATP most of the time to movement. Okay, so this is exactly what happens here. The hydrolysis of the ATP is what cranks the head of the myosin motor protein, and in exchange, it ends up in pressing or in creating this contraction movement, and creating this contraction movement that causes our muscles to contract. And very similar to this function, we also saw cargo proteins or molecular motors that ship or move cargoes on the highways of the cells, which are the microtubules that we talked a little bit about, or the cytoskeleton of the cell. And again, they do it in the same way. They do it by hydrolyzing ATP. And these heads are very similar to the heads that you saw before in the case of the motor proteins. And even we said that there are different types of cargo proteins, like you saw in the nice animation movie that I showed you, that do the distribution locally. So these are like the long range, or you can say like the ships or the oil tankers that cross the oceans. And there are a second set of cargo proteins, which they are very similar to what we have in muscles, because they are myosin heads that are carrying it on actin, which is the same fibers that we have. This is myosin, this is actin. So it's the same types of fibers and the same mechanism that helps us move the muscles. These are the proteins that are in charge of the local shipment in the cells of our body. Specifically, I said that the classic drawing of the neuron is wrong. We'll dwell into that today as well, because this is a really misrepresentation of how a neuron looks like. This is more similar to how a neuron looks like, and this is also a very a massive underrepresentation, because you actually don't see the entire axon of the neuron here. So again, these are the dendrites of the neurons, and this is a pyramidal neuron layer 5 in the cortex that is reconstructed, digitally reconstructed on the basis of imaging of that cell. And you can see here the dotted pink line is like the axon of the cell, which is a lot of times, in a lot of cases, much larger than the actual than the dendritic tree. But again, it depends on the type of the cells. But the most classic example is the motor neuron that we have in our that we have in our spinal cord that has that the cell body is located somewhere here, and the end tip of the axon is located in the tips of our toes. So it's like a meter, or a meter and something long. For like very high people, it's more than a meter long. And but this is just the classic example because this is the neuron that's very easy to measure. But actually nowadays we know that even in our central nervous system, like in our brains, we have single neurons that have axons that are meters long, because they distribute, they innervate huge regions of the brain. So their action is not that straight and linear like the motor neuron, but it's very elaborate but still very long. Okay, and for that reason, not only I think, but a lot of the people in the community now think that the motor neurons and the shipment of proteins and other molecules that we're going to talk about today, so for that function, the motor neurons are very important, specifically in neurons. So then we finished, you see, we didn't do a lot. Then we finished by talking a little bit about purifying, detecting, and characterizing proteins, like the methods. These are like the classical methods. Okay, today we have more advanced methods, but first you need to know like what are the classical methods that are still used today in order for us to define or study proteins in general. So the first step is to purify the protein, and you can purify the protein or separate it according to a length, mass, binding affinity, or specific ligands that bind to that protein, for example, if it's an enzyme or something like that. So one of the most common methods to separate proteins according to their according to their size or the length of the protein is with an SDS gel, or SDS polyacrylamide gel. And we talked about this molecule, the SDS molecule, that through van der Waals interactions, because they're very non-specific and can attach, can bind to any or can be created in any type of molecular, like the van der Waals interaction can happen between any type of molecule. So in this case, because this molecule is linear and flexible, it very easily creates van der Waals interactions, and these interactions compete for the native interactions of the protein and cause it to denature. Okay, to denature into the linear form. And they don't only denature it, they also cover it with a negative charge. And this negative charge makes sure that we have a charge to mass ratio that is constant. So if we now take all our proteins and we pass them through a homogeneous or a constant like electric field, and we don't put any resistance to them, they will all travel in the same speed. Okay? But like if we take them here and we just pass them through a homogeneous electric field without any resistance, they will migrate in the same speed because the charge to mass ratio is constant between, like, it doesn't matter if the molecule is short, then it has more, it has less negative charges, and if it's long, it has more negative charges, but the net force that each molecule will feel will be the same. So in order to separate, we pass them through a gel. This gel creates like a molecular mesh, and this mesh is actually what slows down the large proteins and slows down less the small proteins. So after we load it, the specific sample to this gel, then we can separate according to the size. Okay, so this is a method for separating according to size. Then we talked a little bit about liquid chromatography. In liquid chromatography, we can really separate according to a lot of different factors. So I'll give you examples of, again, we can separate by size, but in this case, it's the opposite of the electrophoresis that we saw before. Here, in this case, the small molecules will adhere to these gel beads and stick to these gel beads much better than the large molecules. Second example that I showed you is according to charge. Then I also showed you an example that you do it according to antibodies, and I talked a little bit about what is an antibody. And there is also assays that you can separate according to some enzymes that are specific or that are that know how to identify specific products. So if you put the enzyme and we can measure the breakdown of the product, then we can also measure the amount of protein like that. But this is just in general terms. Then we focused on a method of Western blotting, which in the article that you had to read today is one of the main figures is based on Western blot. And what you do in Western blot is basically you start with a SDS polyacrylamide gel. You first separate the molecules by size, and then you transfer them through horizontal transfer, it's called, to a membrane. This membrane like sticks all the proteins to that membrane, and then you can expose that membrane to antibodies. So these antibodies, again, are specific antibodies that we designed against the protein that we want to study. We want to measure the amount of the protein that we have, or we want to see if it's even present there. So you put a primary antibody, it detects the protein of interest, and then you want to get like a readout, so you label it with a secondary antibody that knows how to bind to a primary antibody. The difference between the secondary and the primary is that the secondary is usually marked with some kind of indicator, like a fluorescent indicator, a lot of the times, and then what you'll see in the end is that this band will like glow. And then because you know what's the size of the protein that you expect to find, you can measure like the amount, you can measure the amount of fluorescence, and from that deduce the amount of protein that you had. Okay, so for example, if you're doing an experiment, then you want to know whether your manipulation caused the elevation of that protein, of the levels of that protein, then you want to expect to see that this band will be stronger because you have more proteins. Okay, questions about that? Yeah.
Is there a reason why they don't just make the primary antibody also tagged with?
Yeah, there are actually two good reasons for that. One of them is cost, because you can make, because if you separate between the two, then you can use a secondary antibody for a lot of primary antibodies. Like a secondary antibody can detect a lot of primary antibodies. So for example, you can buy a large batch of secondary antibodies and just change the primary antibodies. So because fluorescent probes cost much more, and fluorescently labeled antibodies cost much more than non-fluorescently labeled. So it makes sense to like buy these in large stocks, and they fit to a lot of primary antibodies. And the second reason is amplification. So each one of these primary antibodies, you don't see it from here, but it actually binds about 4, 5, 6 like of these secondary antibodies can bind to a single primary antibody. So what you get is amplification of the signal. Yeah.
So, but then if one antibody can mark a few other antibodies that mark a few proteins, how can you?
No, I didn't say mark a few proteins.
So every antibody marks one protein.
Every primary antibody is specific to one protein.
Okay, and then, well, binds to one protein.
Yeah.
Okay, and then the secondary antibody can mark a few antibodies.
Yeah.
So how can you use that to quantify?
So I said also before that it's not the best way to quantify. Quantification, you need to know, when you talk about quantification, you need to talk about sensitivity. Like if you have twice as much as protein, then you'll probably be able to see it because it's proportional.
So it's just like a giant.
Yeah, but if you have 10% more, this is not a reliable method to check it. Okay, because sometimes you can have like 5 antibodies binding, 4 antibodies. It really depends on the conditions of the reaction. Also, how dirty it is, if you have like background and a lot of other stuff. So it's considered a semi-quantifiable.
Comparative.
Yeah, it's comparative, and like you see in the article, it's really the best way to use it is like all or none. Okay, if you don't have anything and then you have something. Okay, so we didn't talk about it. Now we'll continue talking about just finishing this chapter, and then we'll go over the questions. So one of the ways to characterize a protein is, and the most accurate way until today to characterize a protein is a very, very old method. It's been around for 50 years, at least 50 years, actually at least 60 years, and that is X-ray crystallography. And what's been done in what you actually need to do, or the challenge of this method, is to take your protein of interest and then create a crystal out of it. And what is a crystal? Crystal is a repetition, ordered repetition of the same molecule, like to create, like when you think about if you ever saw like a crystal of salt, it's very ordered repetition of the same molecule that's organized in 3D. So the main challenge in crystallization is creating a crystal of your protein. But once you have a crystal of your protein, then you can bombard it with an X-ray source or X-ray beam, and then by the diffraction pattern, for example, this is one protein that's in charge of unwinding DNA, but it doesn't really matter, and you get this diffraction pattern, and the mathematics behind this diffraction pattern has already been solved 60 years ago. So we know by this diffraction what is the location of each atom of that protein according to this diffraction pattern. So a lot of time when you see like that, we know like in the atomic resolution like the structure of a protein, it comes from these studies. But you need to understand that one of the main criticisms behind it is a lot of time that the protein that you crystallized is not behaving the same way when it's in a crystal like it's in the native form. Okay? Because the crystallization is a very artificial process. For example, Ada Yonat got her Nobel Prize for crystallizing the ribosome, and at least part of the ribosome, the bacterial ribosome, and her patent or her breakthrough in that term is that she used oil. She used like a very stable ribosome, first of all, and second of all, she used oil instead of water in order to crystallize it. Okay, that all the other attempts before her used water for the crystallization. Again, because the assumption is that you don't want to make, like, proteins are supposed to be in water, so you don't want to. I talk all the time about the importance of water for the tertiary structure and all the interactions. So people have avoided using oil or other substances to crystallize things. So she not only crystallized it, but more importantly proved that it's the correct structure. So for that she got the Nobel Prize for that. So actually this is a method that's gaining popularity during the last few years because it's converted from a low throughput to a high throughput method, and I'll explain in a second how. So it's called mass spec or Malditov. Actually, I need to put a title here. But so in mass spec, what you actually do is that you bombard the sample that's sitting on a metal plate with an ionizing laser. So the laser charges the molecule with a positive charge, and then because there's an electric field through these very strong electric fields through these plates, then the molecule accelerate through the space, and the speed of acceleration is inverse proportional to the size or the weight of the molecule. For example, this is much more accurate. You get a much more accurate weight discrimination than what you would get in a polyacrylamide gel. Okay, for example, like this is so accurate that you can actually decipher if you take your sample and use it nowadays, like what really revolutionized this method over the last, I think, 10 years, is that the resolution of this method is so accurate that you can actually identify like 1,000 different proteins and the relative amount from this type of experiment, because it's so how does exactly this is an example of how like a mass spec profile for different proteins would look like. So most of the time the proteins will not stay in their third structure form. This abuse normally breaks them down to smaller pieces, but these small pieces are very predictable. So every protein will break down into its own signature profile of pieces. So this is exactly like the profile that you see here. For example, this is an antibody that was measured with the mass spec, and these are other types of proteins. So you see that each type of protein will have its own signature mark. Okay, so again, nowadays you use this method because it's so accurate. You use this method to quantify. You can quantify 1,000 proteins simultaneously, because you have softwares that can identify the signatures of each one of them, and discriminate between them, and even know the relative amount that you have from each one of these proteins. Okay, so this is a very high throughput method for protein detection. Okay, so questions regarding this chapter? They're not regarding what we talked about right now. So, chaperones are involved in proper folding of proteins. Mark the correct sentence for most proteins in the cell. Again, I'm saying most because there are deviations in biology. So, A, chaperones fold proteins to a thermodynamically unstable shape. Someone thinks yes? No. Chaperones fold proteins into a shape to which the protein is unable to acquire spontaneously. Chaperones promote proper folding. Okay, and chaperones slow down proper folding. No, they actually speed up proper folding. Okay, so an enzyme active site could include a ligand binding site, a DNA binding site. We actually didn't talk about that, but it could. Binding site for specific proteins, catalytic site, hydrophobic site. Right, all of the above. So regarding the hydrophobic, we said a lot of time that the catalytic site of the enzyme is often on the in the internal parts of the enzyme, and we said that a lot of time this will be an hydrophobic site because it will turn inwards. Passing a protein mixture through a standard SDS polyacrylamide gel is a technique of protein separation by which feature are the proteins separated. So charge, right? No. Okay. Size? Yeah, we talked about it just a second ago. Okay. So, moving forward to the incredibly interesting article that you had to read. So, first of all, what did you think about the article? What?
I didn't understand most.
Okay, that's good. So there's a lot to. That one of the challenges of this article is like to separate the ikar from the tafel. Okay, like to look at the more important stuff and the least important stuff. A hint for that is, for example, a lot of the figures or a lot of the things they talk about in the main text are only appearing in the supplementary. So this is like one of the indications that a lot of the things that appear in the main text are not very important. And so you're asking why they're there. It's probably because the reviewers that looked at that paper asked for it to be in the text, and the authors didn't really want to put it, but it ended up there. But it's okay if you thought that this article is boring, because then, hopefully, after we finish talking about it, you'll see that you're wrong, which is a good thing. Okay, so first of all, I give you guiding questions regarding this paper in order to also guide you to the things that are important in this paper and the things that are least important. So, first of all, is an MDA the molecule endogenous? Like, is it naturally found? So where does it come from? You know? Yeah, it's actually synthesized as something that, like a lot of the things in biology, it's a molecule that people have tried, and then they identified after they started synthesizing this molecule that it activates a specific type of receptor, and from that the NMDA receptor actually got its name from. So is an NMDA? So I actually jumped to 3. So what actually makes it open in the body? So the real answer is glutamate, not glycine. Like glycine, yeah, so glycine together, but actually glycine is most of the time there. Okay, most of the time glycine is already there in physiological conditions, and the glutamate is the variable thing. So it doesn't mean that we'll see here in the article that they used artificial conditions where they had glutamate and they added glycine, and then you can see the effect of glycine, but the physiological effect is through glutamate. And is it, we didn't talk about ionotropic and metabotropic, I'll just skip it. What ions enter through an NMDA receptor? So calcium, and like we will learn in this class also, and you'll learn in the rest of your biological life, is that calcium does everything in the cell. Okay, everything is controlled by calcium. So the fact that that calcium enters here is very important. And is it deactivated rapidly or slowly? Slowly compared to what? To non-MDA receptors, which are like normal AMPA or glutamate receptors, which are very common. And this feature is extremely important. Okay, like the deactivation, we'll talk about it, in the end. But just remember that this is very, very important. Is it heteromeric or homomeric? Heteromeric, and this is one of the things that the article showed. And what is the research question? It's not really clear. The research question is, what is the structure, okay, of an NMDA? What is GluR2? No, it's not a subunit. So GluR2 is a receptor, is a subunit of the AMPA receptor. Okay, it's a non-MDA. It's the subunit of a non-NMDA receptor, or the glutamate receptor. And why is it important here? Because when this paper was published, the structure of GluR2 was known. Okay, and this returns to, again, when I described crystallography, then we'll return to that and understand why they always keep repeating to GluR2. Okay? Okay, so the rest we'll answer in the end. So, yeah, we talked about that. So, what did they actually crystallize? Did they actually crystallize the entire protein, or they crystallized only a part of the protein? A part? Which part? Hint. Only the S1, S2 part of the protein. They actually show here how they artificially cleaved it from the mature protein, and they only crystallized these parts. They didn't crystallize these parts and these parts. Okay, so only the binding sites for glycine and glutamate. And they also added linkers here to stabilize the structure. Okay, because they cut it down, so they had to stabilize it later. Okay. Why did they do the crystallography? To know the structure. Before that, before this paper, we didn't know the structure of NMDA, of the receptor NMDA. We just knew the structure of GluR2, but it's a different. Why did they cut it and not?
I mean, did they do the same thing that you talked about before about crystallization? Doing the.
Yeah. Okay. So they crystallized in two ways actually. They crystallized it once when these two are connected to one another, and one of the questions actually that they ask here, and I'm not talking about, is whether they bind, like the NR1 and NR2 are bound like this in the form of homodimer or heterodimer. They found that it's a heterodimer, and so they precipitated these two bound to one another, or in their native form, and they also precipitated only this part, which when it's bound, no, actually only this part when it's bound to glutamate. So they did two precipitations. Okay, so one of the nice things to do in papers, especially when you start reading them, is looking at the editor summary, and to understand the importance of the paper and also to summarize the results. But so obviously the editors take all this part in order to emphasize the importance of it, and this is actually the achievement of this paper. But it doesn't get into too much of the conclusions of this paper. But I can tell you that looking back, this paper has an enormous amount of citations, relatively. For example, the structure, it has 700 citations since 2005. And and the paper, for example, that described the GLUR2 before that had like 200 citations until now. That described it in 2004, I think. So this is a very fundamental paper, and I hope I can convince you why. So the first thing that they did in this paper, so they crystallized it, the structure. So okay, great, you crystallized the structure. So now, what did they need to prove? Like, the first thing is to show you the conclusions of their structure. And the conclusion is, first of all, the structure itself. Okay, so they present the structure and they say, these are the structures and these are the sites that they analyzed as important for in the interaction of the NR1 and NR2A subunits. So first of all, they proved here that it's a heterodimer and not a homodimer like before. So the NR1 and NR2 are bound together or interact together through these three sites. And that also most of the interactions is what they call hydrogen bonds or salt bridge. And salt bridge is actually a combination of two bonds that exist together, for example, in this group. So you have also a simple electrostatic ionic interaction between the negatively charged group and the positively charged group, but you also have a hydrogen bond between the oxygen and the hydrogen here. So a salt bridge is a type of bond that combines hydrogen and electrostatic interaction. Okay? So they show us the structure, and then, so for you, maybe you're satisfied, okay? But for people that know crystallography, they say, wait, okay, let's see if the structure makes any sense. Okay, you showed us structure. We don't know that you crystallized it correctly, or that. So what the authors did here for the first time is said, look, we know that it's very similar in the sequence of the amino acid and to the GLUR2 receptor. We already know how the GLUR2 receptor looks like, so let's see how similar it is. So the answer that they got is the ideal case. Why? Because it's similar enough for us to believe that this is really the structure, because they show that their crystallography, like at least parts of the protein, for example, domain 1 of the protein or the homodimer is very similar to the GLUR2. So the GLUR2 is in pink here, and the green and the blue is their crystallography. So this is for us to show us, yeah.
But just because it's similar to GLUR2, why does that mean that it's right?
Because GLUR2 is already known, and we know that the sequence of amino acid is very similar to one of them. So we know from the if the sequence of the amino acid is similar, so we also deduced, because the tertiary structure is completely, and this time this is the quaternary structure, is influenced directly from the sequence of the amino acid, then we would expect, or the people that study this protein would expect, that it will look pretty similar. But why I say that they got the ideal answer? Because it's not only similar, it's also different. Okay, so in one domain they convince us that they did a good job in crystallizing it, and the other domain they say, wait, but it's not the same, because we know that there are differences between them. What?
But they said that, right? So maybe that is.
Maybe, that's a good question, but they'll answer that in a second, okay? So, what you're talking about is another level of criticism, which is good, but we'll get to that. So, first of all, it makes sense. That's what they want to show you in the first figure. We did a crystallography, it makes sense, okay? But also a small thing that I wanted to show you, this is actually from the supplementary, but it's not a main thing of the article, but it's a nice biological phenomenon, is that they also solved on the way the unique property, or not really unique, but it's a special property of NMDA, that it binds glutamate and NMDA in the same place. So this is one of the things that we always say, that enzymes and proteins are very specific to the type of molecule that they bind to. So, but in this case, we have NMDA and glutamate, which are pretty different from one another. Like they're similar in some ways, but they're different also in a lot of other ways. But so they settled the questions, or to how exactly this binding occurs. And actually what they show here is that when it's bound to glutamate, there are water molecules here that actually, they don't really show it well in the figures, but if you see this W2, a water molecule, when NMDA is bound, then this W2 is out of this cleft, or this binding cleft. And this is what actually helps. So it can either bind water with glutamate or NMDA without the water. So the water, you can say, is like the compensate of the binding of the glutamate. In the case of the NMDA, the water is like kicked out of the place. And they also show here, again, by comparing to GluR2, why NMDA cannot bind to GluR2, because there is a steric interference from this thing. Okay? So this amino acid in this position, in the GluR2, will have a steric clash with the electron cloud of the NMDA, and thus will not be able to bind to it. Okay, so for me it's pretty, again, it's a very strong proof to be able to explain the binding of these two different molecules and why the NMDA cannot bind to the GluR2. So back to our main story. Is the structure real? Okay, so they said, like you said, you crystallized it, maybe the linker is caused the difference, you only crystallized a part of it. So what they did here is that they say, okay, so our structure predicts that in domain, this time we'll go back to in site 1 and site 3, we have close proximity of two amino acids that if we take these amino acids and now substitute them with cysteine, which is a special amino acid, like I remind you that if two cysteines meet each other in close proximity, they will form a covalent bond between them, which is an SS bridge. Okay, so they say, let's say if our model is true, then we will take the physiological. Now they're not basing it on the structure. They base the location of the amino acid according to the structure. And this is how they picked the candidate amino acids, and they mutated them now in the physiological protein, not in the cut protein or anything else. They took the real protein, they expressed it in cells, and what they did is that they show four types of experiment, and this is a Western blot experiment. So as you can see here, this is a size. And what they show here is that when they put only the wild type, the unmutated, and what they mean here is that one of them is like wild type first is NR1, and wild type second is NR2. So when you have the two wild type subunits that are not modified, then you see one band here when they use the anti-NR1. When you see anti, it means antibody. Okay? So antibody against, here they used an antibody against NR1, and these are the bands they detected. So they saw a band here, and they saw a band here. Again, when they did a substitution between, when they just mutated one side and added a wild type in the other side, they still just saw a single band. But what happened here? Can someone explain? What happened? Why is there a higher band here in the mutant-mutant? So what will this mutation cause? This mutation, they added cysteines in locations that they think will be in close proximity according to their 3D structure for the NR1 and NR2A. So if their model is correct, then it means that these two cysteines should be in close proximity enough to create the SS bonds. So what will actually happen is that these two subunits will bind together. And if these two subunits bind together, they're larger, because they now make a larger protein. Because I remind you, in the SDS polyacrylamide gel, you denature the protein, so proteins that are like just linked together through non-covalent interactions will just break apart. And this is what you see here in all the other, but only here you see this higher band, and this higher band represents the binding of these two subunits together through a covalent bond because of the manipulation that the experimenters did in this molecule. And to prove it even further, they add what we call a reducing agent. And this reducing agent, actually what it does, it breaks down disulfide bridges. So this is what it does. It breaks down disulfide bridges. And you see that after they added this reagent, they didn't get this band anymore. So not only that they get this this larger molecule, they also managed to reduce or to get back the original structure when they added this reducing agent that breaks disulfide bonds.
Sorry, what is it confirming? Just where they bind together, the two subunits?
That confirms that their prediction to the exact atomic location of these amino acids and these amino acid is correct. If they were wrong, and their crystal structure was just artificial, and in the physiological terms it wasn't true, then they will not get this band, because like one amino acid would be here and the other one would be here. Right. And they mutated them. And it won't do anything, because they won't bind to one another. So this is actually like a proof that this structure is real. And as you can see, they don't really use it as a quantifiable measure, like the Western blot here. It's again, it's used like an or a none, and also you can see this Western blot is pretty messy. Okay, there are much nicer Western blots. But to prove their point, it's enough. Because here you see a band, here you don't see a band. Okay, so the next thing they did is that they tested like the electrical activity or the electrical, the changes in current in the cell that that happen when they add glutamate in the presence of glycine. So glycine, again, this is like physiological conditions. Glycine is abundant, so it has enough glycine. Glycine are already bound to this receptor, and they added glutamate. And because, again, what you would expect is because the protein has to undergo a conformational change. Okay? So why did they do this? Why did they do this experiment? Because in biology, if there is binding, there is conformational change of the protein. And if you add these anchors to the protein, it will probably, also they predicted from the structure, it will probably affect the ability of the protein to undergo conformational change. So it will screw up the protein, in essence. So what they actually show here, to make a long story short, is that when you add the DTT, which breaks down the disulfide bridges that they did, it almost has no effect. It has a little bit effect, but they explain it. If you really want, I can explain this also. But there's a very small effect on all the different combinations, but only when you have the two subunits that are mutated, hence they have a disulfide bond between them, then the effect of adding the TTT is dramatic on the return to activity of that channel, or the NMDA channel, or the NMDA receptor. Okay? So they show it in both ways. They show it like proving the structure or the size, and proving that the function is restored after they added the DTT. Okay, so now that they actually showed you the structure, and they proved that the structure is real in physiological terms, so what can they learn from that structure? So again, what they did here is that they compared it. They compared the structure to glutamate, and they actually found that there's a single amino acid, there's tyrosine, that is sitting in a location in one of the locations that is different from the GluR2, and it's very similar to what would happen if we would take GluR2 and add this drug to it. So aniracetam is a drug that is known to increase or slow down the GluR2 receptor. So this is known in advance that when you add aniracetam, it's actually been trialed for Alzheimer. Now it's already not trialed for Alzheimer. It's the FDA doesn't approve this drug anymore. You can only buy it in Europe. I don't know why, but it probably didn't work like all the Alzheimer drugs. But what it actually does is that it slows down the activity or the time constant of the GluR2 receptor. So actually, what they discovered here is that the NMDA receptor, what makes it slow down is that it kind of imitates, so the native conformation of the NMDA receptor is similar to what you would get if you would take GluR2 that is bound together with this drug. So it's like a biologically slowed down version of GluR2 that did it in a very similar way that you would get by using this drug. So this is the first thing that you can learn from this structure. And then, okay, so if this is actually correct, what will happen if we swap this amino acid, the tyrosine, or this aromatic loop of the tyrosine, that is sitting here and stabilizes these structures with different amino acids. So what they actually find out is that everything fits their model. That's why it's also a really amazing article, because everything just fits perfectly through the whole steps. So what they did is actually they mutated it. They saw that when they mutated this amino acid, and this is a very good illustration of what happens in the end, their model that they proposed, is that when they mutated this amino acid with a different amino acid that doesn't have this aromatic ring, or carbon aromatic ring, then the kinetics really sped up. So this is the wild type, and you can see that when they added glutamate, they did an assay when they measured the speed of decay of this double exponential. So never mind this, it just shows what you can see here, is that when you compare it to the wild type, when you mutate it to serine, you get actually a much faster closing time of this receptor. Okay? So I know that I'm taking time. I'll finish the next class before time, but it's important to finish this point. So what they actually show is that when you don't have an aromatic ring, in the cases of serine and leucine, which is this one and the black one, you get a faster kinetics. So in essence, this single amino acid is what modulates the activity of the NMDA receptor. And what's even more interesting is that they show that when they add more aromatic rings, like in the case of tryptophan that has 2 aromatic rings here, they actually slow down even more the activity or the kinetics of NMDA. And actually, the type, just a side note, that the types of bonds that stabilize this structure is van der Waals interactions of this ring with the two sides of the with the two subunits in the sides, which is actually very smart, because van der Waals is relatively weak, so it like stabilizes it, but not too much for it to open. If it was like a hydrogen bond or something like stronger, then it will either form or break. So the van der Waals interaction here is like also very distance dependent, so you can understand why it like assumes this conformation, but then breaks down gradually. And also we can, a lot of time we'll talk about it when we talk about DNA, is also that these types of like planar molecules with an aromatic ring are very good for van der Waals interactions and stacking interaction, because of their like their molecular shape and electron cloud. So why is this article interesting? Okay, first of all, because they did a really good job, okay, in showing, like if you look at it in a perspective of critical scientists from their field, you can now understand that they did a very good job in proving their model, and they showed also the physiological aspect of that, is that they not only created models, they generated predictions regarding their model, and they proved it. And they didn't do it on any type of molecule. The NMDA molecule is extremely important for synaptic plasticity and memory. And what is more important is the kinetics of NMDA. Probably the NMDA is important because of its kinetics. Because it's so slow, it actually permits. I'm not going to. Did you learn about NMDA? Yeah, like in this year? No. So you're supposed to learn about it in Bal's course or D. Huh? So it's one of the most interesting proteins in regard to synaptic plasticity, and what is interesting about it is the kinetics and the fact that it's a coincidence detector, like that only when a presynaptic cell and the postsynaptic cells were activated in close proximity with one another, then it will open and allow the permeability of calcium. But the time that it remains open allows for more calcium to enter the cell and create more changes in the cell. So just think about the fact that, and we know actually from experiments that are based on these articles, what happens physiologically when you start mutating, when you start testing these mutants in like animal models and test their memory and test their ability And for example, in some of the cases, certain memory aspects improve if you slow down the NMDA receptor, and certain aspects get ruined when you speed up the NMDA receptor. So it's actually a very, very important. So what I like about this article is that it goes from the level of the molecule, from the atom, to behavior. Okay, and the implications of that is very, very significant to the field. Hence the number of citations that this article has. So it's very important and interesting. Okay, so we'll take a break. היי. אז צריך להסתכל על כאילו בפירוט על המאמרים. יש כל מיני, הבעיה שיש הרבה מאמרים גרועים שנעשו כאילו עם כל מיני מוטציות שעשו ל-NMDA ודברים כאלה, אבל נגיד אסוציאציות או כל מיני דברים כאלה לפעמים השתפרו, כל מיני דברים כאלה שחושבים שצריכים לקרות נגיד בזמן מסוים, אז זה כאילו הזמן של קונסולידציה של נגיד זיכרון או משהו כזה, אז הדברים האלה נגיד יכולים כאילו להשתפר אם אתה כאילו מגדיל את הטווח זמן שהרצפטור הוא אפקטיבי כאילו. אז אבל מה שכן קרה זה שכאילו קורה בעקבות הדברים האלה, זה לפעמים מקבלים יותר אפילפסיה, מקבלים יותר סיז'רס, כאילו, כי יש אובר אקטיבציה כאילו ואובר פעילות ודברים כאלה. אבל כן, הבעיה היא בתחומים האלה שבגלל שאנחנו לא כל כך יודעים לעשות ניסויים התנהגותיים בבעלי חיים, כי הם נורא בייס כאילו, וכל פעם אתה בודק רק את השפיץ, כאילו את הניסוי ההתנהגותי שמתאים לך, אז המאמרים שגם פורסמו על הדברים האלה, זה נורא קשה לדעת כאילו מה נכון ומה לא, כי זה מבוסס, כאילו, נורא קשה להניח התנהגות כאילו, או ללמוד התנהגות ממשהו שאתה משנה משהו כל כך דרמטי. אז כאילו יכול להיות שיפור במשהו אחד, אבל בדרך כלל זה ירוס גם משהו אחר. אז כן, הרבה פעמים זה ככה. אבל כן. האמת ששווה, לא הסתכלתי הרבה זמן, צריך להסתכל על הציטוטים של המאמרים, כל הזמן זה עולה, של המאמר הזה, ולראות כאילו מה חדש בעניין הזה, כי זה כל הזמן משתפר. מה שאני בדקתי שנה שעברה, זה לא היה מאוד מרשים, אבל זה כאילו, אבל היו אפקטים, כן. 2 minutes, madam. I know that I said I'll give you in the end, but I saw that you're enjoying yourself in the coffee break, so I didn't want to. So I decided moving the time to the beginning. But if you're already here, so. Okay, so we can wait. Okay, so this chapter, which is originally titled Basic Molecular Genetic Mechanisms, but I just decided to drop the basic because it's condescending a little bit. It's actually not basic. We learn new things about the basic molecular mechanisms all the time. So in this chapter, which will be pretty brief, we're gonna continue in our zigzag in molecular biology. Like, I know that we just learned about proteins, but now we will return back to the level of the RNA and specifically how DNA turns into RNA and then also protein. So this will be a brief chapter. I know that for a lot of you it's very basic, but it's only going to be this class. And we're also going to address it in the direction of neurons again. So the basic dogma of biology is DNA, RNA, the DNA transcribes into RNA, then RNA translates, DNA transcribes into RNA, and then RNA in turn translates into protein. Okay? So this is a central dogma of molecular biology, and just wrote here, what is a dogma? Dogma in Greek is that which one thinks is true. Okay? So if this is what 20 years ago people looked at RNA as an intermediate step between DNA and protein, like even there are some descriptions that show DNA, then something, and converts to protein. Nowadays we know that this part is very significant, and we'll talk a lot about the functions of RNA as well. And because I study, and our lab studies a lot of RNA, then we'll have also a little emphasis on that. So in general, the role of nucleic acids, and when I mean nucleic acids, I mean DNA and RNA, is first information storage in the form of DNA, which is long-term storage, you can look at it, and mRNA is like short-term storage. So mRNA is messenger RNA that follows by the central dogma, more or less. Like, it's the mRNA that transferred it. It's the messenger of the information from the DNA to the level of the protein. Okay? It also has roles as sequence interpretation. So tRNA or transfer RNA are RNA molecules that know how to identify sequences in the RNA and match the amino acid to them. So they're actually in charge of the translation, and we'll talk about that. rRNA have a structural role. They're part of the ribosome. I told you that the eukaryotic or our ribosome is composed of about 80 different proteins and 5 different RNA molecules. So these RNA molecules are special. They're called rRNA. They're also located in different areas of the cell. And they also have a role, which is very recent, not very recent actually, but it's a exploding field that studies the regulation of expression and even the catalytic activity of what we call non-coding RNA. Okay, so only one type of RNA is what we call coding RNA. When we say coding, it's coding to a protein. And there are three other rows of RNA that we know of. That this is what we call non-coding RNA. So this is RNA that stops in that the functional level is this, and it's not like a messenger of the message from the DNA to the level of the protein. Okay, so you can have either long non-coding or short non-coding RNA, and we'll also talk about them, but in the last chapters we'll talk about them in length. So this is like the chain of nucleic acids, or it describes again like a typical cell. And this is the nucleus of the cell, and this is the cytoplasm of the cell, or the cytosol of the cell. And this is a place where DNA can replicate if the cell is dividing, for example. This is the only case that we describe where we know that DNA is replicating like in the normal life of the neuron, for example. There is no DNA replication. Okay? Yeah.
In the previous slide, which type is the one that we use for junk DNA?
So this. Okay? So, but we're going to talk about it in length. Okay? Like in the seventh topic of the course, it's specifically dedicated to this junk RNA. Okay, so if we're talking about neurons, it's important to know that neurons generally do not replicate. Beside like a subclass of a niche in the hippocampus and the olfactory bulb, where you have newborn neurons, like adult newborn neurons, but in general, after the age of 3 or 2, you have no new neurons in your brain, and what you have is what you're stuck with. And even worse than that, they're dying all the time. So, but the upside is that there's more connections. So you become, you can say like you become smarter, but your potential is decreased. And this is a lot due to the fact that you don't have any replication in the neurons. You don't have duplication in neurons, so they don't multiply. So this is the only place, only when cells multiply, like in your skin or in other tissues that regenerate, then you have duplication of DNA. So inside the nucleus, this is where the transcription and the level from the DNA to the RNA happens. It also happens in steps, which we're not going to talk about exactly now, but we will talk about them in following classes. After the production of the mRNA, like the processing and the production of the mRNA, it comes out of the nucleus and translates in the cytosol. Okay? So again, you can see that this is a very biased view, like it's very like the mRNA is just a step along the way. It's only existent in this part, and then it immediately translates into a protein, and it doesn't have any function beside that. There's a place in the nucleus that's called the nucleolus, which you can actually see in electron microscopes, where the RNA is being produced, like the RNA that is that is part of the ribosome. Okay, it actually has different sets of proteins that know how to produce this type of RNA. It's not that it looks like there's no DNA here. There is DNA. It's like the same, but it's just a separate part of the nucleus. Okay, so tRNAs that again just appeared here, they also transcribed in the nucleus into functional RNA, and then the ribosome and the tRNA together are in charge of translating the mRNA to a protein. Okay, so this is the very general scheme of the central dogma in biology. A little bit about the structure of nucleic acids. So nucleic acids have directionality, much like in the protein where we said that if we have N to C, in nucleic acid we have the 5' end and we have the 3' end. So these 3 and 5 come from the direction or the carbon that is on the backbone of the amino acid of the nucleic acids. So this direction is attached to the 5 carbon of the phosphate of the sugar of the nucleic acid, and this end attaches to the to carbon number 3 of the sugar of the nucleic acid. What you don't see here is the bases, so they're just depicted here as C, A, and G, but there are also bases here, and the structure and the bond that binds 2 nucleic acids together is a phosphodiester bond. Okay, so it's like oxygens with a phosphate in the middle. This is a more like detailed structure of the same molecule of DNA, and this time you have also the reverse strand. So the orientation of the 2 strands is anti-parallel. Okay, it's not in like in when we learned about in beta sheets that you can have parallel beta sheets and anti-parallel beta sheets. In the case of DNA, it's always anti-parallel. Okay, it's always from 3 to 5 and then 3 to 5 like in the opposite direction. And here you can see also the hydrogen bonds that are in charge of binding of the bases. So in general, between G and C, this is the stronger bond that has 3 hydrogen bonds, and between A and T, you have 2 hydrogen bonds. So this is why like the connection between A and T is weaker than the connection of GC. A lot of time you'll find people referring to the term GC content, like in genes, and what they will mean normally is that this area is more tightly bound because of the GC. If it's rich in GC, then the bond between the 2 DNA strands will be stronger. Okay, so the structure of the DNA is what we call like it's a coiled helix, and it has a minor groove and a major groove. So it's like the structure is like you have like a large opening and a small opening, and this repeats itself. And the sugar phosphates or the sugar backbones are facing outwards. Okay, a lot of time people mix up between this structure to the structure of proteins, in which the backbone of the helix is like in the center, and then the amino acids are pointing out. Here, first of all, it's a double helix because it's composed of 2 strands. But in this case, the backbone is pointing outwards, and the bases are pointing inwards. So, like I said, it's anti-parallel, and blah 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 blah. Yeah, we said A and T, G and C, and the bonds besides the hydrogen bonds, the bonds that stabilize the entire structure and make it like coil in the shape that we see it, are hydrophobic and van der Waals interactions that stabilize the structure. Okay, so and a lot of these structures are actually van der Waals interactions that occur. You don't really see it here, but here it's actually depicted much better. You can see that there's like stacking. So the yellow ones are like the bases, these bases. So you need to imagine them as being not like this, like you're seeing here, but like this orientation. Okay, so they're planar and they're like stacking on top of each other, and the van der Waals interactions very similar to what we saw before in the van der Waals interaction of the NMDA receptor. Is that this stacking between them, the van der Waals interaction between the bases, is what causes this compression of the DNA. Okay, and and again, van der Waals is very important also here because DNA is very flexible. There's a lot of proteins that know how to change the structure of the DNA and open it and unwind it, and etc. So you don't want also this bond to be too strong if you're the designer. So the discovery of DNA is largely attributed to the people that also won the Nobel Prize, which is James Watson and Francis Crick, which you can see here. Watson and I think this is Watson, this is Crick. I'm not sure, but the woman that didn't get almost any credit on discovering the structure of DNA is Dr. Rosalind Elsie Franklin, which probably showed her preliminary data to Crick, and then he stole this idea and went on to make the structure. Like the structure that he said was true in the beginning was actually wrong, and only in in the end, the structure that she offered was the correct one, but she died without receiving a lot of credit for that. So we're trying to give a little bit of that credit back. So we can actually find different types of DNA, but the most common type is bDNA, the one that you saw before. And the difference is that you have different levels of compaction. Or for example, if the cell is replicating, or certain enzymes can modify and like squeeze the DNA and alter momentarily the orientation and the structure of the DNA to be like aDNA and zDNA. But in general, the state in which you will find the DNA in the cell will be in the B conformation. And generally, it's a right-handed helix. Right-handed because we have certain directionality, and it like rotates like this. And the helix make a complete turn every 10 base pairs. So it's about it takes 10 base pairs here to make a complete 360 turn around the central axis. Okay, so there are many proteins, especially ones that are in charge of transcribing the DNA to RNA and modulating the DNA, and also regulation factors that know how to bind to the specific structure of the DNA. For example, this protein, which is called Tata box binding protein, which is very important for transcription, it can actually bind to the grooves of the DNA and cause like distort the 3D structure of this and make it like accessible for specific proteins that need to work on this DNA. And it knows also how to bind DNA through this interaction. The DNA is actually stored in our cell in a very, very compact way. So each one of our cells, if we take the DNA and we stretch it, we will get to 1.8 meters long. So if we take all our DNA, in general we have 23 pairs of chromosomes in humans in every nucleus, and they're actually not structured in the. I think I thought I put here a picture of chromosomes, but it's very important to understand that they're not. If you ever saw a picture of chromosomes, I'm sure you have, like these structures that look like I don't know, X's with extended legs or something like that. Then it's not like that in the cell. If the cell is replicating, then there's one stage in which the chromosomes take that shape, but in all other cases, and specifically in cells that are not duplicating like neurons, the chromatin is filling the entire nucleus. Okay? And at first we also thought that this filling is random, but nowadays we know that it's not random, which is pretty. Again, it's crazy, the level of complexity, because you would think that, okay, so you take this multiple strings. Again, when you think about it, a nucleus is like size of 5 to 10 microns in size. It's very small, and you fit in 1.8 meters inside in a non-compact way, not compact like in chromosomes and dense like in chromosomes that you normally that you usually see, and also that the location of each one of these strands relative to one another is not random. Okay, so you have like these hotspots. For example, genes that are important for specific functions, a lot of time will be in close proximity, in 3D close proximity to one another, in order for them to function together and to be regulated together. And this is just one of the things in the past 5 years that people are starting to realize. In general, in biology, every time we think that something is random and doesn't have a reason, in the end we find out that it has. And it's not random. So, okay. So, again, in every cell in our body, we have a lot of DNA that is stacked in there. It is stacked, although you see like this, it's like an open conformation, not in the chromosomal conformation, like packed conformation, but it's still packed around nucleosomes or histones that wind the DNA and keep it relatively ordered. So it's not like a free strand that is like in space. It is packed with these nucleosomes or histones that know how to pack the DNA coil. So the basic structure of a gene is, again, this has changed in the last 10 years or 20 years from a region of DNA that encodes a functional protein. So in the last 15 years it was added also RNA product, because in the past the gene was, because people didn't think that RNA is very important, so again a gene was defined to be something like a DNA region that encodes for a functional protein. That was it. Okay, but now we know that there are also genes that encode for RNA products, the entire non-coding RNAs that fall into that. But it's important to understand that, first of all, everything that you see here, if you hear the word promoter, enhancer, 5' untranslated region, which is UTR, regulatory sequences, or terminators, enhancers, silencers, all these terms, they just mean sequences of DNA. Okay? They're not proteins, they're not anything else. They're just sequences of DNA that we know have specific functions. For example, promoters are something that we use a lot, for example, in neurobiology, to target specific types of cells. Because if we have a cell that we know that I don't know, pyramidal neuron layer 5 expresses Tie1, like a certain type of protein which is Tie1. So we know that if we take the Tie1 promoter and we put some type of gene afterwards, then we can cause the expression of that gene only in the specific types of cells that we're interested in. So the promoter sequences are very specific to the type of gene that they're found before, and there's also specific type of proteins that know how to identify these promoter sequences. Okay, and I know that it's a little bit complex and we're going fast, but we'll repeat these concepts along the semester. So from the level of the DNA, what you actually get in the level of the mRNA is just the 5' untranslated region, 3' untranslated region, and a polyadenylation tail, and the coding region. The coding region, again, is what translates into the functional protein in the end. So the steps of transcription that translate, like, I don't want to say translation because it's like the transition from the level of the DNA to the level of the mRNA happens, again, we divide it into steps, but it's actually, again, it's not steps, it's a continuous process that you can divide also into sub-steps. But in general, and this is also this is an insane underrepresentation of what actually happens here. There are a lot of different factors that are involved here, and a lot of different types of proteins that are involved, but you only see the promoter. So what a promoter actually does here is that this is called RNA polymerase, and there are different types of RNA polymerases, but not very much. So what it does, it actually binds to the promoter region, then looks for initiation for a transcription start site, and from that moment on it will unwind one of the strands of the DNA and will start transcribing an RNA that is complementary to one of the strands of the DNA, and it will be almost identical to the strand that is not transcribed. Okay? So because it's in the reverse orientation and it's transcribing the 3, you see here that the blue one, there's a white strand and a blue strand. So because it's transcribing from like sitting on top of the 3 to 5, the molecules that are gonna come out here, or the RNA molecule strand that is going to come out here, will be first the 5' end will be the first part to be made, to be produced on the basis of the DNA. Okay, so what RNA polymerase does is that on the basis of the template DNA, it produces a complementary nascent or complementary mRNA molecule on the basis of that. Okay, and again to remind you of the directionality, the 5' end and the 3' end. So the connections are almost always. I always say almost because in a few years maybe it won't be correct, but it's always made from like the addition of new nucleotides. For example, in this chain will always be on the 3' end. Okay, so new nucleotides are going to be added in this direction. Okay, so the first part of the molecule that comes out is the 5' end, and then bases are added to the 3' end. And then after the RNA polymerase has finished running along the molecule, it, by mechanisms that I must say are not completely clear to us until today, like we know how it happens in bacteria, but we know less about how it happens in eukaryotes. There are certain models, and they're still debating about that. And again, when you look at it, it's pretty amazing that we still don't know what causes the RNA polymerase to fall off. For example, I can tell you that one of the things that students always mix up is that they think that that if this is the reading frame, or this is the part that codes for the protein, then the RNA polymerase just falls off here. But it doesn't. Okay? It continues throughout this, and it continues even much more. Okay, much more than what you see here. And then just along the processing of the mRNA, the end part is cut or clipped out, and then it's being added a poly A tail. But again, we're going to talk about that in length. So luckily for you, you don't have to imagine too much. I also have a movie showing that, and I know that I went through it pretty fast for people that don't have the basis. But again, this is one of the things that you need to read about in the book if you really want to understand.
Central dogma of molecular...
Okay.
The central dogma...
So what I like about this movie is that it shows everything in real time. Okay, so it shows really the time constants of how it happens. And just imagine that in the cell, if I would have to imagine like how things really look like, I would say that this movie is pretty accurate. But apart from the fact that everything here is much more filled with a lot more DNA and proteins and nucleotides and everything else.
Of molecular biology, DNA makes RNA, makes protein. Here the process begins. Transcription factors assemble at a specific promoter region along the DNA. The length of DNA following the promoter is a gene, and it contains the recipe for a protein. A mediator protein complex arrives carrying the enzyme RNA polymerase. It maneuvers the RNA polymerase into place, inserting it with the help of other factors between the strands of the DNA double helix. The assembled collection of all these factors is referred to as the transcription initiation complex, and now it is ready to be activated. The initiation complex requires contact with activator proteins, which bind to specific sequences of DNA known as enhancer regions. These regions may be thousands of base pairs distant from the start of the gene. Contact between the activator proteins and the initiation complex releases the copying mechanism. The RNA polymerase unzips a small portion of the DNA helix, exposing the bases on each strand. Only one of the strands is copied. It acts as a template for the synthesis of an RNA molecule, which is assembled one subunit at a time by matching the DNA letter code on the template strand. The subunits can be seen here entering the enzyme through its intake hole, and they are joined together to form the long messenger RNA chain snaking out of the top.
Okay. So, yeah, the thing that was not accurate about this is the termination. Again, because we don't know. But we're pretty sure that what happens is that RNA is not being cut out of the ribosome, just that I can tell you what are the models that think what we think about today. What happens in eukaryotes is that there is kind of a torpedo model actually that says that there are certain enzymes that know how to identify regions in the end of the mRNA, and then they cleave this region when the RNA polymerase is still bound, and then there are special types of 5' exonucleases that know how to degrade RNA very fast, and they run along, and they it's called a torpedo model because they're targeting the RNA polymerase, and when they're catching it, they're causing it they cause it to fall off the DNA strand. So this is one of the mechanisms that people think is true, but again, there's much more to do in this field. Okay, so the structure of RNA. So most of the cellular RNA, now we moved on from the level of the mRNA, we moved on to describe other types of RNA. Specifically, the structure of RNA, it's always single-stranded. In most of the cellular RNAs that we find, it's not double-stranded. And it also assumes, very similar to what we saw in proteins, it assumes it has secondary structure and also tertiary structure. A secondary structure in an RNA molecule is much less defined than what we talked about in proteins, and it's simply complementary of bases. Like the bases know how to attach each other to cling to one another, so you can have complementary and all sorts of hairpin and these structures, or what we call stem loop regions of the RNA. This is an example of a tertiary structure, and this is a different way to show the same molecule. And this is a tRNA molecule that, again, this molecule is in charge of pairing codon or a sequence on the mRNA molecule to the appropriate amino acid on the other side of the tRNA. That's why it's called a transfer RNA. So then we move on to one of the most common roles of RNA, which is actually inside the ribosome, or to make proteins. So this process, which is called translation, is actually the translation of the code on the mRNA by the tRNAs that know how to bind to specific triplets on the mRNA molecule of letters, know how to bind specific amino acids. And then actually it happens in this direction. So you have a tRNA that's bound to a specific amino acid that has a specific 3-letter combination that we call an anticodon in this case. This is a codon on the mRNA, which is divided into triplets, and an anticodon that knows how to bind to these codons. And this is all done in the context of the ribosome. And after these tRNAs are binding to the mRNA, then there is a transfer of the amino acid that the tRNA is carrying to the next, to the growing polypeptide chain, or to the protein that is being made. And here we actually know exactly what are the stop and what are the initiation sequences, what are the stop sequences, and how exactly this happens. So again, when we're talking about the different types of RNA, so here we saw the tRNA, the rRNA, and the mRNA are involved in the process of making new proteins. For this reason, for example, people think that RNA is something that is more ancient, that preceded even DNA, because we see that the RNA is the one that has the function. So it makes more sense to us in evolutionary terms that RNA just created DNA along the way in order to store the information probably to longer terms, or because DNA is much more stable than RNA, than the other way around. Then DNA just happened and then spontaneously made RNA that was functional. It was probably the other way around, in which RNA was the first, because we see that RNA also has function. RNA can act also as an enzyme. So for example, in this process, everything here is RNA, except the proteins that support the ribosome. But you can also see examples in lower life forms in nature in which RNA does everything, including cleavage of of other mRNA or RNA molecules. Okay, so in very briefly, the genetic code, which is if you ask me, one of the big mysteries also of life, because this code is actually, and this is in a 3D matrix that is biological or biologist friendly, okay? Because biologists, we don't like 3D, we don't like more than 2 dimensions. So in order to show a 3D matrix, we showed it in 2D. So in order to read this, you actually need to see, like, first of all, the 2 first letters point you to the specific cube, and then the third letter is you can get it from here. So each one of these 3-letter codes in the mRNA mean a specific amino acid, and also we see that there is a redundancy between the different codons. For example, these 4 different codons code for the same amino acid. Okay, meaning that if on the mRNA molecule we will see these 3 codons, we will see all these different codons, they will actually recruit different types of tRNAs, because each tRNA will have its own anticodon region, but all of these tRNAs will be carrying leucine and to this specific location. So this is actually the translation process that translates this code on the mRNA to the level of the protein. And you can see that we have 64 codons or codes that just translate into 20 amino acids, so you have a lot of redundancy. And also there is a very good reason for that, probably, but we're not going to get into that too much, but I can give you a hint that it's because of the third letter, but I'm not going to say more than that. Specifically, we have 4 special codons. One of them that is a start codon. The start codon, actually, every protein will start with this codon. Okay, so on the mRNA molecule, in a certain location, what will happen is that the tRNA molecule, a specific tRNA molecule that has the anticodon that is complementary to this sequence, will go along the molecule, bind to a specific location, bind to this codon to the AUA, and recruit methionine, like the amino acid methionine, and from that point on, the ribosome is going to actually assemble around this amino acid, around this tRNA, and start producing the protein from that moment on. So it's not that like the start portion of the mRNA is not the start portion, is not the first place of the protein. And we also have stop codons, in which when the ribosome arrives to one of these stop codons, what will happen is that there is no tRNA that knows how to identify or to bind to these codons. And what happens is that eventually there is a releasing factor that will bind instead of the tRNA and will cause the whole complex to open and the protein to be released from the ribosome. Okay, so this process, in contrast to what I told you about transcription in the past, translation is pretty much understood. And here you can see like a nice illustration of exactly what happens. So this is the ribosome. The mRNA is like passing through it like a tape. So that's like the best example for that. And the tRNAs are coming one by one and transferring their amino acid according to their binding according to the sequence over here, to the codon sequence over here, and they transfer their amino acid to the growing polypeptide chain. And like always, you don't have to imagine too much. And you have a movie also about translation.
The job of this mRNA is to carry the gene's message. The job of this mRNA is to carry the gene's message from the DNA out of the nucleus to a ribosome for production of the particular protein that this gene codes for. There can be several million ribosomes in a typical eukaryotic cell. These complex catalytic machines use the mRNA copy of the genetic information to assemble amino acid building blocks into the three-dimensional proteins that are essential for life. Let's see how it works. The ribosome is composed of one large and one small subunit that assemble around the messenger RNA, which then passes through the ribosome like a computer tape.
It assembles around the start codon.
The amino acid building blocks, that's the small glowing red molecules, are carried into the ribosome attached to specific transfer RNAs. That's the larger green molecules, also referred to as tRNA. The small subunit of the ribosome positions the mRNA so that it can be read in groups of 3 letters known as a codon. Each codon on the mRNA matches a corresponding anticodon on the base of a transfer RNA molecule. The larger subunit of the ribosome removes each amino acid and joins it onto the growing protein chain. As the mRNA is ratcheted through the ribosome, the mRNA sequence is translated into an amino acid sequence. There are 3 locations inside the ribosome, designated the A site, the P site, and the E site. The addition of each amino acid is a 3-step cycle. First, the tRNA enters the ribosome at the A site and is tested for a codon-anticodon match with the mRNA. Next, provided there is a correct match, the tRNA is shifted to the